For decades, humanity believed it understood viruses. We learned how they spread, how they mutate, and how to stop them. We built vaccines, developed treatments, and declared victories that reshaped modern medicine. But somewhere along the way, scientists realized something deeply unsettling: that one virus was never trying to win quickly. It was never trying to shock the world. It was waiting. This virus does not kill fast. It does not announce itself with chaos or collapse. Instead, it embeds, adapts, and survives inside the very system designed to destroy it. It turns time into a weapon and patience into strategy. And the most disturbing part is not that we failed to stop it, but that stopping it may never have been possible. Tonight, this is not a story about panic, or outbreaks, or headlines. This is a story about what happens when biology outgrows human expectations. About a virus scientists understand better than almost any other and fear because of it. Because the greatest threat is not what we don't know, it's what we already do. The most dangerous biological threats in human history have rarely announced themselves with violence. They did not arrive suddenly, kill instantly, or collapse societies overnight. Those kinds of threats are visible. They provoke fear, urgency, and reaction. Humanity is evolutionarily tuned to respond to immediate danger. What we are far less equipped to recognize are threats that move slowly, quietly, and patiently, threats that blend into normal life before they are understood as dangerous at all. In medicine, speed creates clarity. When people fall ill quickly, when symptoms escalate rapidly, when mortality spikes within days, cause and effect are easy to identify. The danger feels real because it is obvious. Slow threats, however, do not follow this pattern. They create distance between exposure and consequence. They dissolve the emotional link between action and outcome. And when that link disappears, so does urgency. This is why many of the most consequential biological events in history were underestimated in their early years. They did not look catastrophic. They looked manageable. They looked temporary. They looked like problems that could be addressed later. By the time later arrived, the threat had already reshaped behavior, health systems, and entire populations. From a scientific perspective, loud threats are inefficient. They burn brightly, but briefly. They trigger defensive responses, research funding, behavioral changes, and political attention. Quiet threats operate under a different logic. They minimize disruption while maximizing persistence. They allow societies to function normally, while the biology continues uninterrupted. This is not coincidence. It is selection. Evolution does not favor drama. It favors success. And success, in biological terms, is measured not by destruction, but by endurance. A virus that incapacitates its host too quickly limits its own opportunities. It shortens chains of transmission. It isolates itself through fear. It invites containment. In contrast, a virus that causes mild, delayed, or ambiguous harm spreads freely. It benefits from social interaction, travel, work, and routine. It thrives in the very behaviors that define modern life. This is where human intuition fails. We associate danger with intensity. Biology associates success with balance. A threat that allows its host to live, function, and interact is far more likely to persist than one that overwhelms immediately. Waiting is not weakness. Waiting is strategy. The concept of waiting in biology does not mean inactivity. It means operating below thresholds, below pain, below panic, below detection. It means allowing time to dilute responsibility. When consequences appear months or years later, they are attributed to age, stress, environment, or coincidence. The virus does not need to disguise itself. Time does that work naturally. Medical systems are optimized for events, not processes. Acute illness fits neatly into diagnosis, treatment, and resolution. Slow biological harm does not. It accumulates across appointments, across specialties, across years. No single moment demands attention. Instead, patterns emerge only when viewed at scale, and even then, they are easy to debate. This creates a structural blind spot. What cannot be easily measured is often undervalued. What cannot be clearly attributed is often dismissed. And what unfolds slowly is almost always underestimated. History repeatedly demonstrates this pattern. The greatest long-term impacts on human health did not come from sudden pandemics alone, but from conditions that reshaped life expectancy, productivity, and quality of life over decades. These were not moments, they were eras, and they began quietly. Scientists understand this difference instinctively. This is why epidemiologists and evolutionary biologists worry less about headline numbers and more about trajectories. Not how many people are sick today, but how many will never fully return to baseline tomorrow. Not how deadly a pathogen is immediately, but how it alters health over time. The danger of quiet threats is not that they are invisible. It is that they are visible in ways that do not provoke action. Mild symptoms, recoveries that are incomplete but acceptable, damage that is distributed rather than concentrated. These outcomes do not feel like emergencies. They feel like inconveniences, and inconveniences are tolerated. Tolerance is one of humanity's greatest strengths and greatest vulnerabilities. We've adapted quickly. We normalize conditions that would have once seemed unacceptable. This adaptability allows societies to survive hardship, but it also allows slow harm to continue unchallenged. When a threat becomes part of daily life, it stops being perceived as a threat at all. It becomes background, and background risks are rarely addressed with urgency. This is why scientists pay attention not to what alarms the public, but to what fails to alarm it. Silence is not reassurance. Silence is often the signal that a system has adjusted to something it should not have accepted. The most dangerous biological threats are those that exploit this adjustment. They do not demand response. They wait for fatigue to do their work. They wait for attention to move elsewhere. They wait for resources to be reallocated. They wait for the language to soften. Waiting also exploits time horizons. Political systems operate on years. Media cycles operate on days. Human attention operates on moments. Biology operates continuously. A threat that unfolds over decades is misaligned with every major decision-making system humanity has built. This misalignment creates a gap, and that gap is where waiting threats thrive. There is another uncomfortable truth embedded in this pattern. Slow threats rarely produce villains. There is no single failure to point to, no obvious mistake, no clear moment when a different decision would have changed everything. This makes accountability diffuse and resolution elusive. From a scientific standpoint, this is deeply frustrating. Researchers can identify risks, model outcomes, and warn of probabilities, but warnings about slow harm do not compete well with immediate crises. They sound abstract. They lack urgency. They are easy to postpone. Yet probability, unlike fear, does not fade with time. It compounds. Each year of persistence increases exposure. Each exposure creates opportunity. Each opportunity allows refinement. Waiting threats do not rely on sudden breakthroughs. They rely on accumulation, on marginal gains, on time doing what time always does. This is why the most dangerous biological threats are rarely the ones we fear most at first. They are the ones we fear least until fear is replaced by resignation. The realization often comes late, not when infection spreads, but when recovery does not fully restore, when health systems notice subtle shifts in baseline demand, when productivity changes cannot be explained by economic cycles alone, when patterns emerge that were invisible in isolation. By then, the threat has already succeeded in its primary objective, persistence. Waiting threats do not need to win quickly. They do not need to dominate headlines. They do not need to kill fast. They only need to remain. And remaining is far easier when no one is urgently trying to stop you. This is the foundation upon which the rest of the story is built. Not panic, not catastrophe, but patience. Biological patience operating within human impatience, because the most dangerous threats are not the ones that force humanity to react immediately. They are the ones that allow humanity to wait. One of the most persistent misunderstandings about biological danger is the belief that speed equals strength. In human terms, speed feels powerful. Fast threats shock us, overwhelm us, and force immediate response. In biology, however, speed is often a liability. Evolution does not reward the fastest killer. It rewards the most reliable survivor. And survival, for a virus, depends not on how quickly it harms its host, but on how effectively it uses the host to continue existing. A virus does not benefit from panic. Panic isolates. Panic disrupts contact. Panic invites intervention. The faster a virus incapacitates, the faster it triggers barriers to its own spread. Hospitals close doors. Borders tighten. Behavior changes. Resources mobilize. From an evolutionary perspective, this is resistance, and resistance is exactly what evolution works to avoid. This is why many viruses that cause dramatic illness burn out quickly. They surge, peak, and decline. They dominate attention for weeks or months and then fade into history. Their impact is undeniable, but their persistence is limited. Evolution does not measure success in headlines. It measures success in continuity. A slow virus behaves differently. It reduces urgency. It allows time for normalization. It spreads through routine rather than crisis. Hosts continue working, traveling, socializing, and interacting. Transmission chains remain intact because nothing feels urgent enough to break them. This is not accidental behavior. It is the result of selective pressure favoring viruses that do not provoke immediate defense. From an evolutionary standpoint, The ideal virus is one that causes minimal disruption while maintaining consistent replication. Not enough harm to be eliminated, not enough absence to be forgotten, just enough presence to persist. This balance is rare, but when achieved, it is extraordinarily stable. Speed also introduces risk for the virus itself. Rapid replication increases error rates. Large mutations destabilize structure. Aggressive infection invites immune overreaction. Slower replication allows refinement. It allows incremental change. It allows the virus to explore evolutionary space without collapsing its own viability. This is why evolutionary biologists often describe successful pathogens not as aggressive, but as efficient. Efficiency favors patience. It favors strategies that trade intensity for longevity. In this context, waiting is not passive. It is precise. The immune system reinforces this dynamic. When confronted with an immediate, severe threat, the immune response is aggressive. High inflammation, broad activation, maximum effort. When the threat is mild or delayed, the immune system moderates its response. It shifts toward tolerance. This reduces damage to the host, but it also allows the virus to persist. From the virus's perspective, this moderation is advantageous. A host that survives and functions remains a vehicle for spread. A host that is overwhelmed becomes a dead end. Evolution does not care about cruelty or mercy. It cares about outcomes. Speed also shapes memory. Humans remember crises vividly. They organize systems around them. Slow threats fade into background experience. When symptoms are mild or delayed, they are rationalized. Fatigue is attributed to stress. Cognitive changes are attributed to aging. Declining health is attributed to lifestyle. The virus does not need to hide. It is camouflaged by complexity. This creates an asymmetry between biological time and human perception. Biology operates continuously. Humans perceive selectively. What is not immediately disruptive is rarely prioritized. Evolution exploits this gap effortlessly. The concept of flattening curves reveals this tension clearly. When immediate impact is reduced, success is declared. Yet reduction in speed does not mean reduction in presence. Slower spread can increase total exposure over time. The virus trades intensity for duration, and duration often produces deeper impact. This is counterintuitive to non-specialists. A threat that feels less severe is assumed to be less dangerous. But in long-term population health, slow degradation often causes more cumulative harm than short, intense shocks. The damage is distributed rather than concentrated. It is harder to see, harder to measure, and harder to reverse. Evolution favors strategies that distribute harm rather than concentrate it. Concentrated harm provokes response. Distributed harm becomes background. Another advantage of slowness is flexibility. A virus that moves slowly can adjust to changing conditions. Behavioral shifts, partial immunity, medical interventions. Instead of being extinguished by sudden change, it adapts incrementally. Each adjustment is small, but together they sustain persistence. Speed also limits learning. Rapid outbreaks end before long-term effects are understood. Slow infections generate data over years. This may seem beneficial for science, but it also means the virus has already established itself by the time understanding matures. Knowledge arrives after entrenchment. There is a deeper implication here that unsettles researchers. A virus optimized for waiting does not need to defeat medicine. It only needs to coexist with it. Treatments suppress severity. Vaccines reduce risk. None of these eliminate presence. The virus adapts to the boundaries imposed, rather than attempting to break them. This creates a long-term equilibrium that favors endurance over eradication. The virus accepts limits. It stays within tolerable ranges. It does not provoke extreme countermeasures. This is evolutionary intelligence without consciousness. Speed also exposes vulnerability to competition. Rapidly spreading viruses compete aggressively for hosts. Slower viruses coexist more easily. They occupy niches without overwhelming systems. They persist alongside other pathogens, environmental stressors, and social behaviors. This coexistence increases resilience. From a global perspective, speed attracts attention. Waiting attracts complacency. One generates mobilization, the other generates fatigue. Fatigue is more damaging to long-term response than ignorance. It erodes consistency. Scientists studying long-lived pathogens often describe a moment when urgency gives way to management, when language shifts from elimination to control, when timelines extend indefinitely. This shift does not occur because the threat disappears. It occurs because speed never forced a decisive moment. This is the point where evolution has effectively won its most important battle, not against the immune system, not against medicine, but against attention. The weakness of speed, therefore, is not biological. It is psychological and systemic. Speed triggers defense. Waiting dissolves it. This is why evolutionary success is often invisible in the short term. The most successful viruses are not those that overwhelm humanity briefly, they are the ones that remain quietly present across generations. They reshape baselines, they alter expectations, they change what is considered normal health, and they do so without demanding confrontation. Speed creates enemies, waiting creates permanence, and permanence is the ultimate evolutionary advantage. One of the most dangerous misunderstandings in human history is the belief that speed equals threat. We instinctively fear what kills quickly because it feels dramatic, visible, and immediate. Explosions, fevers, bleeding, collapse, these signal danger in ways the human brain is wired to recognize. But biology does not share our instincts. In nature, the most successful pathogens are rarely the fastest killers. They are the ones that remain invisible long enough to secure their future. A virus that kills its host too quickly limits itself. The moment symptoms become obvious, detection follows. Isolation begins, transmission slows. In evolutionary terms, rapid lethality is often a failure, not a success. The viruses scientists fear most do not announce themselves. They arrive quietly, integrate slowly, and persist patiently, sometimes for decades, before revealing their full consequences. Latency is the defining weapon of such viruses. Instead of overwhelming the body, they embed themselves within it. They hide inside cells, integrate into genetic material, or remain dormant within tissues that the immune system rarely monitors aggressively. During this time, the host appears healthy, life continues, relationships form, families grow, communities expand, and all the while, the virus travels unnoticed. This waiting strategy fundamentally alters the balance of power between humans and disease. Medicine is reactive by nature. It responds to symptoms, damage, and measurable harm. A virus that causes none of these for years or even decades escapes the entire diagnostic framework modern healthcare depends on. By the time signs appear, the infection has already succeeded at its primary goal, persistence. What makes this even more dangerous is that latency is not passive. The virus is not asleep. It is actively interacting with the host's biology at a molecular level. It is shaping immune responses, influencing cellular repair mechanisms, and sometimes subtly increasing mutation rates within infected cells. These changes are often too small to detect individually, but over time they accumulate into irreversible biological consequences. The immune system, despite its sophistication, is optimized for clear threats. It excels at recognizing foreign proteins, destroying infected cells, and remembering past invaders. But latent viruses exploit a weakness in this design. By minimizing protein expression or mimicking host molecules, they reduce the immune system's ability to distinguish friend from enemy. The body adapts to their presence, often tolerating them rather than eliminating them. This tolerance is not a truce. It is a quiet occupation. The virus becomes part of the biological environment, influencing everything from inflammation levels to cellular aging. In some cases, the immune system remains permanently activated at a low level, leading to chronic inflammation. In others, immune exhaustion develops, reducing the body's ability to fight unrelated infections later in life. The waiting virus also benefits from time in another way: evolution. Each replication cycle carries the possibility of mutation. Over years, this creates a vast internal population of viral variants within a single host. Some of these variants may be harmless. Others may resist immune pressure more effectively. A few may gain new abilities entirely. The longer the virus waits, the more chances it has to optimize itself. This internal evolution means that when the virus eventually reactivates or when conditions change, it may no longer resemble the version that first entered the body. Treatments designed decades earlier may fail. Immune memory may no longer apply. The virus has used time as a laboratory. Another reason slow viruses are so dangerous is behavioral. Humans base risk assessment on cause and effect that occur close together. When harm appears decades after infection, the connection is rarely made intuitively. This delay weakens public awareness, reduces urgency, and fragments responsibility. Policies focus on immediate crises, not invisible timelines. Funding follows headlines, not incubation periods. As a result, slow viruses often circulate widely before society even recognizes them as a threat. By the time epidemiological patterns emerge, millions may already be infected. Containment becomes impossible, not because science failed, but because time was allowed to work in the virus's favor. From a purely statistical perspective, waiting viruses reshape populations rather than individuals. They do not overwhelm hospitals overnight. Instead, they increase long-term disease burden, shorten life expectancy subtly, and place sustained pressure on healthcare systems for generations. Their impact is measured not in daily death counts, but in lost decades of productivity, chronic illness, and intergenerational transmission. Perhaps the most unsettling aspect is that waiting viruses do not need to become more aggressive to become more dangerous. They only need to persist. Every additional year inside a host is another opportunity to spread, adapt, and embed themselves deeper into human biology. In this sense, time itself becomes the virus's most powerful ally. Scientists understand this pattern well, which is why the most feared viruses are not those that dominate news cycles for a few months, but those that linger quietly in the background of civilization. These are the pathogens that rewrite public health not through panic, but through permanence. And this is why the question scientists ask is no longer, how deadly is this virus? But instead, how long can it stay? Because in biology, survival is influence. And influence does not require speed, only patience. Section 4. How the virus outsmarts the immune system. By the time scientists realized that some viruses do not rely on speed, they were forced to confront a more uncomfortable truth. The human immune system, remarkable as it is, was never designed for enemies that refuse to behave like enemies. Immunity evolved to fight invaders that announce themselves loudly, bacteria that multiply rapidly, toxins that disrupt cells, pathogens that leave clear molecular fingerprints. Slow, adaptive viruses exploit the blind spots of this system with precision. The immune system works by recognition. It identifies patterns that do not belong, marks them as foreign, and destroys the cells carrying them. This process depends on visibility. Viral proteins must be expressed. Infected cells must signal distress. Antibodies must be able to bind to stable structures. But some viruses learned long ago that the most effective way to survive is not to fight immunity, but to avoid being seen by it at all. One of the primary strategies involves minimizing viral expression. Instead of producing large quantities of viral proteins, these viruses restrict their activity to the bare minimum needed for survival. They remain transcriptionally silent for long periods, generating almost no signals that immune cells can detect. To the immune system, the infected cell appears normal. No alarms are raised. No attack is launched. Other viruses take a more deceptive approach. They actively interfere with immune signaling pathways. They block the presentation of viral fragments on the surface of infected cells, preventing immune cells from recognizing that infection has occurred. Some even manipulate cytokine signaling, dampening immune responses in surrounding tissue and creating a localized zone of tolerance, where the virus can persist unchallenged. This manipulation is not crude, it is highly specific and often elegant. Viral proteins interact directly with human immune regulators, mimicking natural inhibitory signals that the body uses to prevent autoimmunity. The immune system, designed to avoid attacking itself, hesitates, and in that hesitation, the virus survives. Another layer of immune evasion involves genetic integration. Certain viruses permanently insert their genetic material into the host's DNA. Once integrated, the viral genome is no longer an external invader, it becomes part of the host cell itself. Every time the cell divides, the virus is copied along with it. From an immunological perspective, this creates a paradox. Attacking the virus would mean attacking the body's own genetic material. This integration transforms the infection from an external threat into an internal condition. There is no longer a clear boundary between host and pathogen. Treatment becomes extraordinarily difficult, because eliminating the virus requires either targeting every infected cell or altering the genome itself, both of which carry catastrophic risks. Even when the immune system does recognize these viruses, the battle is often prolonged and exhausting. Chronic immune activation leads to immune fatigue. T cells become less responsive. Antibody production weakens. Over time, the immune system adapts not to eliminate the virus, but to coexist with it. This coexistence, however, comes at a cost. Persistent immune activation contributes to systemic inflammation, which accelerates aging, damages tissues, and increases susceptibility to other diseases. Cardiovascular disease, neurodegeneration, metabolic disorders, and cancers all become more likely in bodies that are quietly fighting an unresolved viral presence. The virus may not kill directly, but it reshapes the biological terrain in which other diseases emerge. Vaccines, one of humanity's greatest medical achievements, struggle against these pathogens for similar reasons. Vaccination relies on stable targets proteins that remain consistent across viral generations, but viruses that persist long-term often mutate precisely in these regions. The immune memory generated by vaccination may become obsolete, as the virus evolves within hosts. Furthermore, vaccines are most effective before infection. Once a virus has established latency or integrated into host cells, vaccination cannot remove it. At best, it can reduce reactivation or slow progression. This shifts public health strategy from eradication to management, a fundamentally different and far more expensive challenge. Antiviral drugs face similar limitations. Many antivirals target active replication processes, but a virus that is not actively replicating is largely unaffected by these treatments. This forces patients into lifelong therapy, where medication suppresses viral activity without eliminating the underlying infection. The virus remains present, waiting. Waiting, in this context, is not passive. Environmental stress, immune suppression, aging, or co-infection can all trigger viral reactivation. When this happens, the immune system is often weaker than it was at the time of initial infection. The virus, meanwhile, has had years to refine its survival strategy. This dynamic reveals a deeper problem. Human medicine is built around endpoints: cure, clearance, recovery. But some viruses do not respect these categories. They exist in a state between disease and normalcy, between activity and dormancy. They force scientists to confront the possibility that control may be the best achievable outcome. The realization that a virus can permanently coexist with humanity is profoundly unsettling. It challenges the foundational belief that science, given enough time, can solve any biological problem. In these cases, time is not neutral. Time strengthens the virus. What makes this even more troubling is that these immune evasion strategies are not rare anomalies. They are the result of millions of years of coevolution between viruses and their hosts. The virus is not malfunctioning. It is functioning perfectly. By understanding this, scientists began to see that fear was not irrational, it was analytical. The virus they feared most was not one that overwhelmed the immune system, but one that quietly convinced it to stand down. And once that happens, the question is no longer how to fight the virus, but whether fighting it is even possible at all. Section 5. Why a cure may be scientifically impossible. At some point, every major medical challenge reaches a moment where the language itself begins to fail. For centuries, humanity has framed disease in terms of victory and defeat. A pathogen appears. Science responds. A cure is discovered. The story ends. This narrative has shaped public expectation so deeply that the absence of a cure is often interpreted as a temporary failure rather than a fundamental limitation. But certain viruses force science to confront a far more unsettling possibility, that the concept of a cure may not apply at all. To understand why, it is necessary to examine what a cure actually means in biological terms. A true cure requires complete elimination of the pathogen from the body, leaving no remaining genetic material, no dormant reservoirs, and no capacity for reactivation. For many infections, this is achievable because the pathogen exists entirely outside the host's genetic framework. Kill the organism and the disease ends. But some viruses do not exist as separate entities once infection is established. They become embedded. When a virus integrates into the host genome, it crosses a biological threshold. Its genetic material becomes indistinguishable from the host's own DNA at the cellular level. From that moment forward, every attempt to remove the virus risks damaging the very cells required for life. This is not a technological limitation alone. It is a structural one. Eliminating the virus means eliminating parts of the host. Even in cases where viral integration is partial or indirect, the challenge remains. Many persistent viruses establish reservoirs, collections of infected cells scattered throughout the body, that remain inactive for long periods. These reservoirs are often located in tissues that are difficult to access pharmacologically, such as the brain, lymphatic system, bone marrow, or immune-privileged sites. Drugs may suppress viral activity in the bloodstream, while leaving these reservoirs untouched. The existence of reservoirs means that viral clearance cannot be confirmed through absence of symptoms or even absence of detectable viral particles. The virus may simply be waiting. This creates a permanent uncertainty in treatment outcomes. A patient may appear cured for years, only to experience resurgence when conditions change. From a scientific standpoint, this undermines the very definition of cure. Another fundamental obstacle lies in mutation. Viruses that persist long-term generate extraordinary genetic diversity. Unlike human cells, which possess robust error correction mechanisms, many viruses replicate with high mutation rates. This allows them to explore vast genetic possibilities over time. Any therapeutic intervention becomes a selective pressure, encouraging the survival of resistant variants. This evolutionary flexibility means that even highly effective treatments may never achieve permanent elimination. the virus adapts faster than the treatment paradigm evolves. What begins as a cure becomes a management strategy. Suppression replaces eradication. The immune system, often viewed as the ultimate backup, faces similar constraints. Immune memory depends on stable targets, but viruses that continuously mutate or alter protein expression erode the reliability of immune recognition. In some cases, immune responses themselves become part of the problem. Chronic immune activation damages tissues, accelerates aging, and creates conditions favorable to further viral persistence. There is also the issue of timing. Many persistent viral infections are acquired early in life, long before medical intervention is possible. By the time treatment begins, the virus has already established deep biological roots. Erasing decades of integration and adaptation is not equivalent to treating an acute infection. It is an attempt to reverse biological history. Gene editing technologies have been proposed as potential solutions. In theory, tools capable of identifying and excising viral genetic material from host DNA could offer a true cure. In practice, this approach introduces unprecedented risks. Targeting integrated viral sequences requires extraordinary precision. A single off-target cut could disrupt essential genes, trigger cancer, or cause irreversible organ damage. At scale, such risks become unacceptable. Even if perfect precision were achievable, another problem emerges: delivery. Editing every infected cell across the entire body is a logistical impossibility with current or foreseeable technology. Missing even a small fraction of reservoirs allows the virus to re-establish itself. In this context, almost cured is biologically meaningless. This leads to a difficult conclusion that many scientists are reluctant to state publicly. Some viruses are not problems to be solved, but conditions to be lived with. They are not temporary invaders, but long-term biological occupants. Medicine can reduce harm, extend life, and improve quality of living, but it may never be able to completely remove them. This realization reframes the entire relationship between humanity and disease. The question shifts from how do we cure it, to how do we coexist with it without collapse? This is not a surrender. It is an acknowledgement of reality. Public discomfort with this idea is understandable. The concept of incurability conflicts with cultural expectations of progress and mastery over nature. But biology is not obligated to align with human narratives. Viruses evolve under different rules, guided by survival rather than morality or intent. What makes this especially unsettling is that scientific impossibility does not require technological failure. It emerges from the basic architecture of life itself. DNA integration, cellular dependency, immune tolerance, these are not flaws. They are features of biology that viruses have learned to exploit. As researchers came to accept this, fear began to take on a different character. It was no longer fear of catastrophe, but fear of permanence. A virus that cannot be cured does not need to overwhelm society to change it forever. It only needs to persist. And persistence, as biology has shown repeatedly, is often more powerful than destruction. Section 6. The laboratories racing against time. Once scientists accept that a cure may be biologically unreachable, the entire purpose of research shifts. Laboratories around the world are no longer chasing eradication. They are racing to slow, contain, and outmaneuver a biological process that never stops evolving. This is not a battle measured in breakthroughs alone, but in margins: how many years can be gained, how much damage can be delayed, how long stability can be maintained before biology adapts again. Inside these laboratories, time is the most unforgiving variable. Every experiment is conducted against an opponent that replicates faster than any human institution can respond. While research proposals take months to approve and clinical trials take years to complete, viruses mutate continuously inside millions of hosts. The scientific method, rigorous and cautious by necessity, is fundamentally slower than viral evolution. Researchers begin by mapping the virus at the deepest molecular level. They analyze its genome, its proteins, its replication machinery, and its interactions with host cells. But unlike simpler pathogens, this virus does not rely on a single critical weakness. Its survival strategy is distributed across multiple systems. Blocking one pathway rarely stops it. It simply reroutes its behavior through another. This forces laboratories to adopt combination strategies. Multiple drugs, multiple targets, multiple layers of suppression. Each added layer increases complexity, cost, and risk of side effects, the body becomes a battleground not just for virus and immunity, but for competing pharmacological interventions that must coexist long-term without destroying the hosts they are meant to protect. Even when promising compounds are identified, translation into real-world treatment is slow and uncertain. What works in isolated cells often fails in living organisms. What works short-term may fail over decades. Persistent viruses expose a weakness in medical research. Most trials are not designed to measure outcomes across a human lifetime. Meanwhile, laboratories must also confront variability. No two infections are identical. The virus behaves differently depending on genetics, age, immune history, co-infections, nutrition, and environment. A therapy effective in one population may be ineffective or harmful in another. This forces researchers to abandon the idea of a universal solution and move toward personalized management strategies, dramatically increasing complexity. Another layer of urgency comes from viral reservoirs. These hidden pockets of infection resist nearly every known intervention. Researchers develop increasingly sensitive detection methods, only to discover that reservoirs are more widespread than previously believed. Each discovery expands the scale of the problem rather than shrinking it. Immunologists attempt to retrain the immune system, enhancing its ability to recognize infected cells without triggering self-destruction. This requires extraordinary precision. Too little activation allows the virus to persist. Too much activation causes autoimmune damage. The margin for error is narrow, and the consequences of miscalculation are severe. Virologists explore ways to lock the virus permanently into dormancy, preventing reactivation, even if elimination is impossible. But dormancy is not static. It depends on cellular conditions that change with stress, aging, and disease. A strategy that works in youth may fail decades later. Laboratories are forced to think not just in years, but across entire human lifespans. At the same time, global coordination becomes unavoidable. No single laboratory can address a virus that circulates worldwide. Data sharing accelerates discovery but also exposes uncertainty. Conflicting results emerge, hypotheses rise and fall. Each setback reinforces a sobering reality. Scientific progress is not linear, and time does not pause while consensus forms. Funding pressures further complicate the race. Persistent viruses do not generate the same urgency as fast-moving outbreaks. They lack dramatic peaks. Their damage is gradual, diffuse, and often invisible in the short term. Convincing governments and institutions to invest continuously over decades requires political will that biology does not wait for. Within laboratories, researchers are acutely aware of this imbalance. They are not racing toward a finish line, but against erosion of immune function, of public attention, of institutional patience. Each year without a definitive solution allows the virus to entrench itself further in the global population. Technological advances offer hope, but also reveal limitations. Artificial intelligence accelerates pattern recognition in viral evolution, yet predictions remain probabilistic, not certain. High-throughput screening identifies thousands of potential compounds, but only a fraction prove viable. Precision medicine advances rapidly, but scalability remains elusive. Perhaps the most difficult challenge laboratories face is psychological rather than technical. Scientists are trained to believe that understanding leads to control, but this virus defies that expectation. The more deeply it is understood, the clearer its resilience becomes. Knowledge does not eliminate fear, it refines it. This is why the work continues despite uncertainty. Not because victory is guaranteed, but because delay carries consequences. Every incremental improvement, slower progression, reduced transmission, extended health represents real lives affected. The laboratories are not seeking perfection. They are buying time. And time, in this context, is not neutral. It is the currency that determines whether society adapts faster than the virus does. Each experiment, each trial, each partial success shifts the balance slightly but never permanently. As the research community pushes forward, a quiet understanding settles in. The question is no longer whether science can defeat the virus outright. The question is whether science can stay ahead long enough to prevent collapse. And that realization sets the stage for the moment when confidence, built on decades of expertise, begins to fracture not in ignorance, but in clarity. Section 7. The moment scientists admitted fear. There is a point in every long scientific struggle where certainty begins to erode, not because knowledge is lacking, but because knowledge becomes complete enough to reveal its own limits. For decades, virology advanced with confidence. Each new discovery reinforced the belief that complexity could always be reduced, that mechanisms could be mapped, and that control would follow understanding. But with this virus, something different happened. The deeper scientists looked, the less final their conclusions became. Progress no longer pointed toward resolution. It pointed toward permanence. The first signs were subtle. Research papers grew more cautious in tone. Words like eradication quietly disappeared, replaced by management, suppression, and long-term outcomes. Conferences that once focused on elimination strategies began dedicating entire sessions to containment ethics and lifetime care models. This was not public panic. It was professional recalibration. Scientists were adjusting their expectations, not outwardly, but internally. What made this shift unsettling was that it did not arise from failure. The science was working, data was accumulating, models were improving, yet each advance revealed another layer of viral resilience. Every pathway blocked uncovered alternative routes. Every immune strategy strengthened exposed new forms of immune evasion. The virus was not winning by overpowering science, it was winning by enduring it. In closed-door meetings and internal reviews, a pattern emerged. No single experiment disproved the possibility of a cure, but no combination of results supported it either. The absence of contradiction became its own form of evidence. Scientists began to recognize that they were not missing a key insight. They were confronting a biological reality shaped by evolution far older than modern medicine. This realization carried emotional weight. Many researchers had devoted entire careers to this problem. They had trained students, built laboratories, secured funding, and published thousands of pages of data, all with the implicit belief that the end goal was attainable. Accepting that it might not be required a form of professional grief, not despair, but mourning for an assumption that had guided decades of work. Fear entered not as panic, but as clarity. It emerged from simulations that refused to stabilize, from longitudinal studies that showed irreversible damage despite optimal treatment, from population models that predicted persistent global burden even under best-case scenarios. These were not speculative fears, they were statistical ones. One of the most troubling moments came when researchers realized that success itself could mask danger. Treatments that extended life also extended viral presence. Suppression strategies reduced transmission rates but increased the size of a long-term infected population. Every improvement carried secondary consequences that unfolded slowly, invisibly, and irreversibly. This forced scientists to confront a deeply uncomfortable question. What if the virus does not need to evolve further to remain dangerous? What if its current form is already sufficient to persist indefinitely within human civilization? This possibility reframed fear entirely. The threat was no longer mutation-driven catastrophe. It was equilibrium. Equilibrium, in this context, meant a stable coexistence where the virus remained embedded in humanity at a constant, unavoidable level. Not explosive, not dramatic, simply permanent. For a scientific community trained to respond to crises, this was profoundly destabilizing. How do you mobilize urgency against something that does not escalate? The fear deepened as researchers recognized that public perception lagged far behind scientific understanding. Society continued to view the virus through the lens of outbreaks and emergencies, while scientists increasingly saw it as infrastructure, something woven into demographics, healthcare systems, and life expectancy itself. This disconnect created ethical tension. How much should the public be told? How do you communicate a threat that has no clear endpoint? Some scientists argued that transparency demanded honesty, even if it undermined optimism. Others worried that acknowledging incurability could weaken prevention efforts or fuel fatalism. This debate did not play out in headlines. It unfolded in advisory panels, peer reviews, and policy drafts. Fear here was not of the virus alone, but of the consequences of truth. As younger researchers entered the field, they encountered mentors who spoke differently than their predecessors. The language shifted from conquest to stewardship, from solutions to mitigation. This generational transition reflected a deeper change. Science was no longer preparing to end the problem, but to live with it responsibly. The most sobering admissions occurred when experts compared this virus not to past pandemics, but to geological forces, slow, shaping pressures that alter landscapes over centuries rather than destroying them overnight. In that comparison, fear was not dramatic. It was existential. The virus was not an event. It was a condition. This recognition also altered how scientists viewed their own role. They were no longer solely problem solvers. They were custodians of long-term stability, responsible for managing a biological presence that could not be removed. This responsibility carried weight. Every recommendation affected millions of lives across generations. Fear intensified when researchers considered the implications of complacency. A virus that waits thrives on neglect. Reduced funding, reduced surveillance, reduced vigilance all create opportunities for resurgence. The absence of visible crisis does not indicate safety. It indicates patience. What finally unified the scientific community was not a single discovery, but convergence. Independent teams, using different methods across different continents, began reaching the same conclusions. This convergence did not inspire confidence. It inspired sobriety. When uncertainty collapses into agreement, fear becomes rational. By this point, the question was no longer whether scientists feared the virus. It was whether fear itself was the correct response. Fear, when grounded in evidence, becomes caution. Caution becomes policy. Policy becomes behavior. In that chain, fear serves a purpose. But the most unsettling realization remained unresolved. If science, with all its tools, foresight, and collaboration, could not guarantee a cure, what happens when the virus changes again? Not dramatically, not explosively, just enough. That question did not belong to the laboratory anymore. It belonged to the future. Section 8. The virus scientists fear the most. For years, the name was avoided not out of secrecy, but out of precision. Scientists understood that naming the virus too early distorted the discussion, collapsing complexity into familiarity. But once the patterns were fully mapped, once the behavior could no longer be mistaken for coincidence, the identity became unavoidable. The virus scientists fear most is not the fastest, not the loudest, and not the most immediately lethal. It is the one that mastered persistence, integration, and patience more completely than any other known human pathogen. That virus is HIV. The fear surrounding HIV does not come from ignorance. It comes from understanding. Decades of research have stripped away myth, stigma, and misunderstanding, revealing a pathogen whose danger lies not in mystery, but in clarity. HIV does not overwhelm the body. It infiltrates it. It does not destroy immunity in a single blow. It dismantles it gradually, systematically, and with extraordinary efficiency. What makes HIV uniquely feared among scientists is not merely that it attacks the immune system, but how it does so. HIV targets the very cells responsible for coordinating immune defense CD4 plus T lymphocytes. These cells are not replaceable components. They are the strategic command centers of immunity. By infecting them directly, HIV turns the immune system against itself, using its own machinery to replicate and spread. Once inside a CD4 cell, HIV performs one of the most consequential acts in virology. It reverse transcribes its RNA into DNA and integrates that DNA into the host genome. This integration is not accidental. It is precise, efficient, and permanent. From that moment forward, the infected cell is no longer simply compromised, it is transformed into a long-term viral reservoir. This is the point at which HIV ceases to behave like a traditional infection and begins to resemble a genetic condition. The virus is no longer an external invader circulating freely. It is embedded within the biological identity of the host. Every division of that cell propagates the virus silently, invisibly, and indefinitely. The immune system cannot remove what it cannot distinguish from self. Antibodies cannot target integrated DNA. T cells cannot eliminate infected cells without destroying essential immune capacity. Medicine confronts a paradox. The more aggressively it attacks the virus, the greater the risk of collapsing the immune system it is trying to protect. HIV's latency is not passive dormancy. It is strategic invisibility. The virus can remain transcriptionally silent for years, producing no proteins, triggering no immune response, and leaving no detectable trace in blood tests. During this period, the host appears healthy. Life continues uninterrupted. Yet the virus is present in countless cells, waiting for the precise conditions that allow reactivation. This waiting behavior fundamentally alters disease perception. HIV does not demand attention. It does not force immediate response. It allows society to forget it exists until it is too late. This is why scientists do not fear HIV as an outbreak. They fear it as an architecture. Antiretroviral therapy represents one of the greatest achievements in medical history. It transformed HIV from a death sentence into a manageable condition. But management is not elimination. These drugs suppress viral replication. They do not remove the virus. The moment treatment stops, viral activity resumes, often within days. This dependence reveals the underlying truth. HIV remains present, active, and capable. The necessity of lifelong treatment introduces a new dimension of vulnerability. It ties survival to infrastructure, supply chains, healthcare access, political stability, economic continuity. HIV does not merely test biology, it tests civilization. Any disruption, war, Poverty, neglect, misinformation creates openings where the virus can regain ground. What intensifies scientific fear is HIV's capacity for variation. Its replication process is error-prone, generating immense genetic diversity within each host. This diversity allows rapid resistance development, forcing continual adaptation of treatment regimens. The virus evolves not globally, but individually, turning each infected body into a microevolutionary arena. Even more troubling is the scale. HIV is not rare. It is embedded across populations worldwide. Millions carry integrated viral genomes in their cells. From an evolutionary standpoint, HIV has already succeeded. It has secured long-term persistence within the human species. Attempts to cure HIV through gene editing, immune reprogramming, and reservoir elimination have achieved remarkable partial successes. But partial success is not cure. Removing 99.9% of viral reservoirs is insufficient. The remaining fraction is enough to restart infection. Biology does not reward near completion. Scientists fear HIV because it exposes a boundary science may not be able to cross, not due to lack of intelligence, effort, or technology, but because of the fundamental rules governing cellular life. HIV aligns perfectly with those rules. It exploits them without violating them. Perhaps most unsettling is that HIV does not need to change to remain dangerous. It does not need to become more lethal. It does not need to spread faster. Its current form is already optimized for persistence in a globally connected species with uneven healthcare access and long lifespans. This is why HIV is studied not just as a virus, but as a warning. It demonstrates what happens when evolution produces a pathogen that values time over destruction. It shows that a virus does not need to defeat humanity to shape it permanently. Scientists fear HIV not because it cannot be treated, but because it cannot be erased. It represents a category of threat that modern medicine was never designed to resolve. A threat measured not in emergencies, but in generations. And once that realization settles in, the question that follows is no longer about HIV itself, but about what comes next, because evolution does not stop at one success. When you are ready, the continuation is Section 9. What happens if it evolves again, where probability, not surprise, becomes the central driver of future risk. Section 9. What happens if it evolves again? Once a virus has achieved persistence, evolution no longer needs to be dramatic to be consequential. The most dangerous changes are not sudden leaps, but subtle adjustments, small shifts in behavior that compound over time. HIV has already demonstrated this principle repeatedly, not through catastrophic mutation, but through incremental optimization. Each replication cycle, each selective pressure, each immune interaction offers another opportunity for refinement. Evolution does not aim for novelty. It aims for survival efficiency. The question scientists now ask is not whether HIV will evolve again, but how. Evolution is not a hypothetical event waiting in the future. It is a continuous process unfolding within millions of hosts simultaneously. Every person living with HIV represents an independent evolutionary environment, shaped by unique immune systems, treatment regimens, co-infections, and social conditions. From a biological perspective, this creates a distributed, global laboratory operating at a scale no human institution could replicate. Most mutations fail. They weaken the virus or render it non-viable. But evolution does not require most changes to succeed. It requires only a few to confer advantage. Over decades, even rare advantages accumulate. Resistance to drugs, altered cell targeting, improved latency control, and enhanced transmission efficiency have all emerged through this slow, relentless process. None appeared overnight. Each arrived quietly, detected only after consequences became visible. One of the most concerning evolutionary directions is refinement of latency. HIV already possesses an extraordinary ability to remain dormant, but latency is not a fixed trait. It can be optimized. A virus that becomes better at hiding, better at synchronizing reactivation with immune weakness, or better at minimizing cellular damage, while persisting, would become even harder to control. Such changes would not increase mortality dramatically. They would increase permanence. Another potential pathway involves immune interaction. HIV's relationship with the immune system is already paradoxical, but evolution favors efficiency. A virus that can further reduce immune activation while maintaining replication would face less selective pressure from both immunity and treatment. This would result in infections that are harder to detect, slower to diagnose, and easier to spread unnoticed. Transmission dynamics also remain subject to evolutionary pressure. HIV does not need to spread faster to become more dangerous. It only needs to spread more reliably. Even minor increases in transmission probability under common conditions could translate into significant population-level effects over decades. This is particularly concerning in regions where healthcare access fluctuates or where prevention strategies are inconsistently applied. Scientists also consider recombination, a process where different viral strains exchange genetic material within the same host. HIV is especially capable of this due to its replication strategy. Recombination allows the virus to combine advantageous traits from multiple lineages into a single genome. This is not random chaos. It is accelerated evolution. The existence of multiple strains within one individual increases the likelihood that new variants will emerge with enhanced survival characteristics. These variants may not replace existing strains globally, but they do not need to. Persistence within subpopulations is sufficient to maintain evolutionary momentum. Another concern lies in treatment-driven evolution. Antiretroviral therapy, while life-saving, creates selective environments that favor resistant variants. This does not indicate failure of treatment. It is an expected evolutionary response. As therapy becomes more widespread, the evolutionary landscape shifts. The virus adapts not to evade treatment entirely, but to tolerate it better. This creates a feedback loop. Improved treatments extend lifespan, increasing the duration of viral host coexistence. Longer coexistence allows more evolutionary cycles. More cycles increase the probability of advantageous mutation. Over time, this loop favors viral stability rather than elimination. Importantly, scientists do not expect HIV to evolve into something radically different. The fear is not of transformation, but of refinement. A virus that is already highly adapted does not need radical change. Small improvements are enough. Population-level modeling reinforces this concern. When long-term persistence is factored into projections, even conservative evolutionary assumptions result in sustained global burden across generations. These models do not predict collapse. They predict normalization. HIV becomes a permanent feature of human health, shaping demographics, healthcare systems, and economic planning indefinitely. This normalization carries its own risks. As urgency fades, surveillance weakens, prevention fatigue sets in, funding priorities shift, evolution exploits neglect. A virus that waits benefits from distraction. Scientists are particularly wary of the illusion of stability. Stable case numbers can mask underlying evolutionary change. The virus may appear controlled while silently improving its efficiency. By the time instability becomes visible, reversal may be impossible. What makes this scenario especially sobering is that it does not require worst-case assumptions. It emerges from realistic probabilities applied over long timescales. Evolution favors patience. Human institutions struggle with it. The future shaped by HIV evolution is therefore not defined by surprise, but by probability. No single mutation matters. What matters is accumulation. Decades of small advantages compound into structural permanence. This realization forces a shift in perspective. The threat is no longer an outbreak, a variant, or a crisis. The threat is continuity, a virus that continues to exist, adapt, and persist without demanding attention, becomes part of the biological background against which all other health challenges unfold. Scientists fear this not because it is dramatic, but because it is predictable. And in science, predictability without control is one of the most unsettling outcomes imaginable, which leads to the final question not about biology, but about humanity itself. Because if the virus does not decide the future through evolution alone, then something else must. Section 10. The choices that decide the outcome. At this stage, biology has already spoken. The virus exists, persists, and evolves according to rules that humanity did not write and cannot repeal. What remains undecided is not the virus itself, but the environment in which it will continue to operate. The future, from this point forward, is shaped less by molecular biology than by collective human behavior. The decisive variable is no longer mutation. It is choice. The most important of these choices concerns attention. Persistent threats do not demand urgency. They erode it. When danger becomes familiar, it fades into the background of daily life. HIV has already crossed this threshold in many parts of the world. It is no longer perceived as a crisis, but as a condition. This shift reduces fear, but it also reduces vigilance. From a biological standpoint, reduced vigilance is opportunity. Public health success creates its own paradox. Effective treatment lowers visible mortality, which lowers perceived risk. Lower perceived risk weakens prevention behavior. Weak prevention increases transmission. This cycle does not indicate failure of medicine. It reflects the limits of human psychology. We respond to spikes, not plateaus. The virus benefits from stability. Another choice lies in how societies distribute resources. HIV does not spread evenly. It exploits inequality. Regions with unstable healthcare systems, limited access to treatment, political conflict, or social stigma create reservoirs of vulnerability. These reservoirs are not isolated. In a connected world, no population remains biologically separate for long. Containment is only as strong as the weakest system. The decision to treat healthcare as a long-term infrastructure investment rather than an emergency response determines whether viral suppression remains continuous or fragmented. Interruptions in treatment are not merely individual failures, they are evolutionary accelerators. Each interruption allows viral replication, diversification, and adaptation. Continuity, not innovation alone, is what restrains evolution. Stigma represents another critical choice point. Viruses exploit silence. When infection is hidden, delayed, or denied, transmission increases. Stigma does not protect society. It protects the virus. Scientific understanding has long recognized this, yet social behavior often lags behind evidence. Fear of judgment continues to delay testing, disclosure, and treatment, extending the virus's reach. Education plays a similarly underestimated role. Knowledge does not eliminate risk, but ignorance amplifies it. Comprehensive, sustained education reduces transmission not through fear, but through normalization of prevention. When education becomes politically inconvenient or culturally contested, the virus gains space to persist. Policy decisions regarding research funding shape timelines measured in decades. Persistent viruses do not align with election cycles or budget horizons. Long-term investment in surveillance, treatment optimization, and prevention infrastructure determines whether management improves or stagnates. Scientific progress without political patience is fragile. Another choice concerns how success is defined. If success is measured solely by reduced mortality, complacency follows. If success is measured by reduced transmission, improved quality of life, and minimized long-term burden, vigilance remains justified. Metrics guide behavior. Behavior shapes outcomes. The global response also depends on cooperation rather than competition. Viruses do not respect borders. Nationalized strategies create gaps. Shared data, shared resources, and shared responsibility slow viral adaptation by reducing uneven selective pressures. Fragmentation accelerates divergence. Technology introduces further choices. Advances in monitoring, diagnostics, and predictive modeling can detect change early but only if they are deployed equitably. Unequal access to technology recreates the same vulnerabilities that allow persistence. Precision without reach is incomplete. Perhaps the most profound choice is philosophical. Humanity must decide whether it views persistent viral presence as a temporary anomaly or a permanent condition requiring stewardship. Stewardship implies maintenance, responsibility, and humility. It acknowledges limits without surrendering effort. This shift challenges a foundational narrative of modern science that every problem has a solution, if pursued aggressively enough. Some biological realities do not yield to conquest, they yield to management. Accepting this does not diminish science, it aligns it with reality. The danger lies not in acceptance, but in resignation. Stewardship requires continuous engagement. The virus does not need humanity to give up. It only needs humanity to look away. History shows that civilizations are not undone by threats they recognize clearly. They are undone by threats they normalize without managing. HIV tests this boundary continuously. It does not force decision through crisis. It waits for drift. The outcome, therefore, is not predetermined. Biology sets constraints, but society sets direction. The virus will persist regardless. The question is whether its impact remains controlled, contained, and minimized, or whether neglect allows gradual expansion. In this sense, the future diverges not at a mutation, but at a meeting, a policy vote, a funding decision, a cultural shift. Small choices accumulate just as mutations do. Over decades, they define trajectories. The most unsettling truth is also the most empowering one. Humanity is not powerless here. It is responsible. The virus has done what evolution designed it to do. What happens next depends entirely on how seriously that reality is taken, because in the end, the virus does not decide the outcome. We do. Conclusion. The last line of defense. After everything science has uncovered, one truth remains impossible to ignore. The virus did not win by overpowering humanity. It won by understanding us, our biology, our behavior, our attention span, and our tendency to relax once danger feels familiar. It learned how to survive not just inside the body, but inside civilization itself. There will be no final battle, No moment of eradication, no single breakthrough that closes this chapter forever. What exists instead is a long responsibility, one that extends beyond laboratories and hospitals into policy, culture, education, and collective memory. The virus will continue to do what evolution trained it to do: persist, adapt, wait. The future will not be decided by whether the virus changes again. It will be decided by whether humanity continues to care once fear fades, whether vigilance outlasts comfort, whether knowledge is matched by action. Because in the end, this virus does not threaten us with extinction. It challenges us with permanence. And history will remember not how advanced our science was, but how seriously we chose to live with what we knew.